Good. In that case, I will start. So, first of all, thank you very much for giving me the opportunity to present the data from work that I've been carrying out in very close collaboration with my dear friend and colleague Steve Moss, who may be on this meeting as well. And it's been quite an interesting journey for us insofar as it started off with very much an exploratory fishing exercise, if you like, that's been proved to be very, very fruitful in the end. And it started because we're clearly interested in dysfunctional vessels in disease, and as you would all appreciate, vessels can either existing vessels can become dysfunctional and end up leaking, and you get then conditions in the retina such as diabetic macular edema. Or you can also get new vessel growth, which is also quite a common feature of many diseases. And almost without exception, new vessels in disease grow abnormally. And as a consequence, they're poorly perfused, they leak, and they hemorrhage. And of course, we see that in two very extensive diseases in the eye, in peripheral diabetic retinopathy and also neovascular AMD, but also in other conditions, including cancer. And we have done quite a large amount of work also on cancer, but I won't talk about that today. But what is fully accepted, of course, is that abnormal vessels, whether they be existing vessels that have become abnormal or new vessels that fail to mature and stabilize, they contribute to the disease pathogenesis. And again, that's not just the case in the eye, it's the case in many, many diseases. And there's been a great deal of success, particularly in the eye, in targeting one of the primary activators of permeability and indeed new vessel growth, and that is the anti-VEGFs. And these have proven to be extremely potent therapies. And of course, we're interested in the diseases in the eye, as I've mentioned, both AMD, neovascular AMD, where you get these abnormal vessels growing, which contributes to vision loss, and also diabetic macular edema, where the existing vessels become dysfunctional, or in the later stages, proliferative diabetic retinopathy, where you get new vessel growth, and these again can hemorrhage and be very destructive. And although the anti-VEGFs have been incredibly successful in the treatment of these diseases, and increasingly now in application in DMO, at any point in time, approximately 50% of patients fail to respond adequately. And so there remains a really large unmet clinical need, and this very much has been shaping our approach to our research. And one of the things that is quite a conundrum, not just in ocular research, but in vascular biology itself, is why under normal situations in development do you get new vessel growth that leads to stable and mature vessels that are functional, but in disease, again, almost without exception, you get either disturbance of existing vessels where they become highly leaky, or in the case where new vessels grow, they fail to stabilize. And this is something that has really puzzled vascular biologists for quite a while. And it begs a question as to whether there are factors that appear in the disease setting that actually might be partly responsible for the failure of vessels to form in a natural way or a physiological manner. And this very much was what was behind our original research when we took a couple of mouse models of retinal disease in which there were abnormal vessels. And we took those vessels and we did a transcriptomic assay on these to see whether there were any differentially expressed genes. And the gene that was most highly overexpressed in these abnormal vessels from these different models, in the vessels, that is, not the rest of the retina, was this gene coding for secreted glycoprotein, leucine-rich alpha-2 glycoprotein 1, and we call this large 1 just for ease. And we have published this back in 2013. And at the time, it was very clear that under normal conditions, nearly the only organ that really produced large 1 was the liver, and possibly also some subsets of leukocytes. But otherwise, it wasn't expressed. But in disease, clearly that wasn't the case from our studies where we took these abnormal vessels from retinal disease. So what we did was to see whether indeed LRG1 was induced in more conventional and commonly used models of ocular vascular abnormality. So we turned to the CNV model, the laser-induced CNV model, and as you can see here, with this RNA scope image in the vascular lesions, you can see messenger RNA in red for LRG1 that has appeared in these lesions and co-localizing with endothelial cells, telling us that indeed this gene is switched on. And if you just look at qPCR of the whole tissue of the retina or the RPE choroid, again, quite a significant upregulation of the gene. In the oxygen-induced retinopathy model, again, widely used in ophthalmic research, but also as a model of angiogenesis itself. During the hypoxic phase of the disease, we also see a dramatic increase in gene expression. And more recently, in some more preliminary data, in 2 models of diabetes, in the streptozotocin model and in the Akita 2 mice, we can see a significant upregulation of message for LRG1. And this is particularly interesting, for example, in the STZ induced diabetes. This is around 2 months, so way before you really see any gross pathology. So it is really quite an early responsive gene in the disease process. Well, what about in humans? Well, we were able to get some vitreous thanks to help with Jim Bainbridge at Moorfields. And we were able to show that just by Western blotting, in this case, there was an increase in LRG1 compared to vitreous from epiretinal membrane patients. And a colleague of ours in Italy has been able to show that was in PDR and in neovascular AMD, a colleague in Italy has been able to show that in aqueous, they also get an increase in the amount of protein, LRG1 protein, in disease. And quite interestingly, if one looks at the literature for those studies that have looked at the vitreous proteome, and there aren't that many of them, everyone we've come across, it's very clear that LRG1 is significantly increased in the vitreous against the controls, whichever controls they used. So this all points to LRG1 certainly being present in disease and therefore may well be contributing to it. An important sort of finding was that early on we were able to generate the LRG1 knockout mouse, and this was, at the time we were a bit disappointed, but actually thankfully it was a great thing to find, was that these mice were perfectly normal and are healthy and fertile, and that's good because it suggests that unlike VEGF, when you knock it out, which is embryonically lethal, LRG1 doesn't seem to have an overt role in normal physiology. What we then decided to look at, see whether we could change the lesion development or alter lesion development in this case CNV if we induced the lesions in LRG1 knockout mouse, and sure enough, both the size of the lesion was reduced in the knockout mouse, and importantly, also the amount of leakage coming from that lesion. Again, using LRG1 knockout mouse and inducing oxygen-induced retinopathy, we were able to show that it didn't affect the normal, more physiological closure of the avascular region, but it dramatically reduced the number of these neovascular tufts that lie at the interface between the retina and the vitreous. So again, this all pointed towards LRG1 contributing to the vascular abnormalities in these models. We then developed, at this time, a polyclonal antibody and showed that intraocular injection of this polyclonal gave a very nice dose response in terms of reduction in lesion size, and that it was equivalent to blocking the VEGF pathway, in this case using antibody against the VEGF receptor 2. And there was some indication that in combination that you actually got a much greater effect. And I could discuss that later, should anybody wish. In the OIR model, what we were able to do is show that if we use suboptimal doses of either anti-LRG1 polyclonal or the anti-VEGF receptor 2, that it was suboptimal such that it didn't have effect. When you put them together, you did elicit a reduction in the number of vascular lesions, again suggesting that there might be some additive effect here. So what exactly is happening? Well, we learned a lot from a lot our tumor work that had suggested that LRG1 is disruptive in terms of altering the endothelial cell parasite interactions, and parasites are critical for the stabilization and maturation of vessels. And what we found was that in the neovascular tufts, in the LRG1 knockout mice, there was much better coverage of the parasites over the vessels, either measured here by alpha smooth muscle actin or NG2 as markers of these mural cells. And this is quite interesting because it does raise the possibility that LRG1, if it's altering the parasite endothelial interactions, may well be playing an early effect in diabetes. And sure enough, again, in some very preliminary data, we have found that one of the earliest things that you can see are these very small focal leaks of the vasculature, in this case in the streptozotocin mice, and in the knockout mice, you see that these are significantly, the number of these are significantly reduced. Well, if our hypothesis is correct that LRG1 is indeed affecting parasite endothelial interactions, what happens if we introduce it during normal physiological angiogenesis of the retina, where, I hasten to add, the gene isn't expressed? And if we introduce the protein, and we've done this also by gene expression, if we introduce the protein into the retina during development, we see a reduction in the amount of overlap and co-localization of parasites with endothelial cells, here shown either by NG2 or PDGF receptor beta expression. So this all points to LRG1 being a disruptive agent in the vasculature, and it of course then sort of raised the possibility that we might be able to generate a therapy targeting this. So what exactly and how does it work? I won't go into the details because it would take a few weeks to go through, but one of the things that it certainly seems to affect is TGF-beta signaling, and it's been known that TGF-beta can signal via two arms depending on the configuration of the receptor complex, either a sort of a more homeostatic arm or a proliferative migratory arm, depending on which configuration is expressed on the membrane. And what we found was that in the presence of LRG1, the configuration of receptors turns towards a ALK1 mediated pathway, activates a certain set of SMADs that drives a more proliferative migratory phenotype in endothelial cells. And this is certainly one of the primary effects, mechanisms of action. We were also able to show that IL-6 is a major driver of LRG1 gene expression, along with a couple of other cytokines, but certainly you can see gene expression increasing when you treat endothelial cells with IL-6. And IL-6 has been reported to be quite disruptive to the vasculature, and some of the disruptive effects are due to, again, alterations in the parasite coverage of the vasculature. And we were able to show that actually what's happening is that IL-6 is switching on LRG1, and it is LRG1 that is responsible for these parasitic effects reported to be due to IL-6. So we have a schematic here whereby we think that IL-6, and again, possibly along with TNF or IL-1, can activate transcription of LRG1. LRG1 is secreted, can either have effect on the endothelial cell itself or on nearby mural cells, and certainly on the endothelial cell, it works through TGF-beta signaling, and this leads to vascular dysfunction. So this, as I say, raises the possibility of a new target. And as I mentioned earlier on, it is really important to identify new targets in vascular problems in the eye, as well as in cancer and other diseases, because often patients will become refractive or non-responsive to the VEGF arm. And that, of course, suggests that there are other pathways that act towards driving vascular dysfunction. And we propose that indeed the LRG1 pathway is a major pathway driving vascular dysfunction in the absence of LRG1. So therefore may well be a very useful target therapeutically. And how have we gone about this? Well, we set off, Steve and I put in applications initially to the MRC to generate, under their DPFS scheme, to generate at the time over 100 antibodies. We ended up with a lead antibody that was the most promising. This was then humanized and deimmunized for human use and hinge stabilized and put on an IgG4 background for various reasons. We then got further funding from the MRC to take this into clinical into the clinic. But first of all, what we had to do is generate master cell bank and manufacture, sort out all the upstream downstream processing, and then test this in NHPs. At this point, we came across a slight problem in that the full length antibody caused an inflammation that was probably not acceptable, and we still to this day are not entirely sure why that was the case. Nevertheless, we decided to take a new tack, and through the formation of a spin-out company with the support of UCL Business and the UCL Tech Fund and Albion Ventures, we obtained seed funding to generate a Fab fragment, which actually has advantages anyway, because it has there are less frequent injections involved because you can get a higher molar load into the vitreous. These were tested in some preliminary experiments in NHPs for tolerability, and we found virtually no inflammation at all. So we were very happy that that our Fab fragment is the route which we go for intraocular treatment. Just this week, we've started the Series A funding to take us into the clinic and do all the manufacturing, and hopefully this will be happening over the next few months. So just finally, it's not just the eye where LRG1 is involved. There's now a growing amount of evidence, and you can see the interest from the publications, the interest in LRG1 is growing exponentially. It's even been implicated in the microvascular pathology of SARS, and it's certainly dramatically increased in COVID patients, but also in many cancers, diabetic kidney disease, and the vascular disease. So on that note, I'd like to thank you for your attention and thank the current bunch of guys in the lab who are doing an absolutely tremendous work, and whose work is some of the work I've shown, and of course my great colleague Steve Moss, who I've done all this work with, and the funders, predominantly the Wellcome Trust and the MRC. And on that note, I'll finish. Thank you.
Okay, thank you very much, John.